Okay, grade 10. This is lesson number 9, carbon and physiology. So we're going to be getting even smaller scale now as we get into this unit, and we're really doing with biochemistry today, bringing in chemistry of carbon and how that is related to biology and connection between, specifically with digestion and homeostasis. So we're now working on big idea number 3. How does carbon move within molecules, cells, systems, and organisms? So you're going to start learning about this today, and we'll give you a talk about later on in the rest of the unit. But I want you to start thinking about what you'd write down for this today, and you're welcome to write down some more ideas for it as well as you go. Here's today's templates. We have the magic of carbon. You're going to do a small diagram here about carbon. Carbon compounds, a bigger diagram, some more complicated carbon compounds here. The 4 macromolecules of life. You can do another diagram here, and here's an assignment using digital mapping software of molecules to compare different carbon compounds. So take a minute, pause the video, and make the template. And again, if you're doing it digitally, make sure you have some sort of template to organize the subtitles, the title, so I can find your stuff really easily in your notes, and you can find it as well. Okay, so let's talk about carbon. So what do you remember about the structure and properties of carbon from our chemistry unit, from last year's chemistry units, from middle school? What do we already know about carbon? Well, hopefully you remember that carbon is a non-metal. It has 6 protons, specific carbon, which has 6 electrons. Big thing here has 2 on the inner electron orbital and has 4 valence electrons. And how many electrons will fill a valence orbital? So it takes 8 to fill it. It's exactly halfway empty. And since it's halfway empty or halfway full, it's pretty stable compared to other carbon compounds, so other molecules. Because it's not too close to being full, not too empty, it's pretty stable. So because it's exactly half full and has 4 valence electrons, this makes it pretty stable and very flexible. So it can do all sorts of different things because it has a space to bond with 4 different atoms. So because of this, it can form single bonds, like here's a carbon with 4 single bonds to 4 different hydrogens to make methane. It can also form double bonds when we talk about methane in our climate change unit. It can form double bonds, like here carbon dioxide has a double bond here and a double bond here. So each oxygen has 2 bonds with carbon, and the carbon tool has 4 bonds, 2 with each oxygen. It can form triple bonds, so here's ethylene with triple bond between the 2 carbons and 1 hydrogen on each side. So the carbon always has a 4 bond total, so 1, 2, 3, 4 for this carbon, and 1, 2, 3, 4 for that carbon, 1, 2, 3, 4 for this carbon, 1, 2, 3, 4 for that carbon. And carbon can also form rings like this, and they make chains of rings. So we have double bond, a single bond, double bond, a single bond, double bond, a single bond. And these rings can be joined to very, very large molecules. We've already talked about some of them, and we'll talk about more about them today. And because of this flexibility and the fact that carbon is found in almost all organic molecules contain carbon, it's just organic molecules, we consider carbon the keystone element for life on Earth. So of all the elements that exist in the periodic table, the one that's key for living things and therefore very important for biology is carbon. Other common elements you might find in organic molecules, organic compounds, hydrogen, oxygen, nitrogen, and phosphorus, the 4 most common ones. We'll see in different compounds today. So they can find those in carbohydrates, lipids, proteins, and nucleic acids, which are the 4 biomolecules we're talking about today. We talked about some of these already in digestion. There are other elements as well you can find, but they're not nearly as common as those 4. Carbon is the most common. And then you have things like iron. Iron's important in your blood cells and hemoglobin proteins. It's important, but it's not nearly as important as carbon, and it's not nearly as common. You find sulfur in some parts of proteins. There's all sorts of molecules and elements involved in organic compounds, but the 4 main ones are carbon, hydrogen, oxygen, nitrogen, and phosphorus, the 5 main ones. Carbon being number 1. So that's what I want you guys to draw, just to remind yourself that carbon can form all these bonds, and that's what makes it so special. So these are kind of a simplified Rutherford diagram, We have the nucleus, and we have 2 electrons being shared here for a single bond. We have 4 electrons being shared for a double bond, and 6 being shared for a triple bond. So single bond, double bond, and triple bond. I didn't put any rings here because a ring is just made of circle of single bonds and double bonds. Okay, let's talk about some examples of carbon compounds. So there's very complex and very simple carbon compounds both that are taken up in the body, that are used in the body. They're broken down, built back up again. Carbon gets rearranged in different molecules throughout different organisms all the time. A great example we've already talked about quite a bit is starch to glucose to glycogen. So you've heard of all 3 of these before. Some of them the last lesson, and some of them earlier. Hope they sound familiar to you. Can you remember what processes, what organs are involved, what's happening with these 3 different compounds? Well, if we start with starch is this really big chain here, that's starch. And it's made up, little tiny bits that make it up, those are glucose molecules. They're called the subunits that make up starch are glucose molecules. So in digestion, amylase, one of the enzymes, it starts in the salivary glands in the mouth. It's going to break down this big starch and start to break it into smaller subunits, into maltose, just like a smaller number of glucose molecules together to make that sugar. And then the small intestine, amylase is also there, produced by the pancreas, to then break it down further. And we have maltase, which also works on maltose to help break it down into glucose. So we have this really big molecule, and we're breaking it down through digestion into single glucose molecules. So glucose is the final product, and glucose is what we talk about for homeostasis. We have a set point for blood sugar, blood glucose level. And we use the glucose in the blood, at the muscles and all the cells, in the mitochondria to do respiration and get energy that we can use. Glucose is it's the carbon compound that we can get energy from the most readily. So we have a lot of it in our bloodstream, but we have a certain amount. Can't have too much, can't have too little. So when we have too much glucose in the blood, do you remember what happens? Well, your liver's going to take it in. Your liver converts it to glycogen. And insulin is the hormone that tells your body to do this. We talked about hormones a little while ago. So insulin is the hormone making your body take in glucose, convert to glycogen. If we go back here, this is what glycogen looks like. It's all these glucose subunits in a different kind of chain. So this has rings and kind of twists in it. It was quite different from the starch chain, but they're both long chains of glucose subunits. Okay, and then if the blood glucose level gets too low, then the hormone glucagon, a different hormone, it's part of that same feedback loop, it's going to make the glycogen be converted back to glucose, so you get that glucose in the blood. So glucose is a subunit for lots of things, not just for glycogen and for starch. It's also in sucrose. So here's a sucrose sugar, which I'm sure you've all heard of. It's a glucose, here's a glucose plus a fructose bonded together. Here's lactose, in dairy products we have a galactose and a glucose. And a maltose is a glucose and a glucose, so 2 glucoses. And this is what's going to be broken down. Amylase breaks starch into maltose, and then maltase breaks maltose into glucose. So what you need to really understand here is that it's constantly, carbon is being taken from large molecules like starch, broken down through processes like digestion, and then built back up again, so that glucose then built back into glycogen for storage. It can also be broken down and used in cell bodies and tissues and a whole bunch of variety of purposes. And lots of other carbon compounds are the same thing. So fructose can be broken down from sucrose. If you break that bond, you get a fructose and a glucose. Galactose, whenever you have dairy, if you drink milk, you're going to get lactose in that. Your body digesting it, you can get glucose from it and a galactose from it. So 2 different sugars there. All these little dots here, little kind of joints between the lines, that's a carbon. So in these kind of diagrams, they don't draw every single carbon and every single hydrogen because it would be really complicated and too hard to see. So if you're taking chemistry next year, you'll get more practice at this. But this little joint here, where there's just a corner and an O, that's a carbon with an oxygen double bonded to it. And that's going to be a carbon, that's a carbon, that's a carbon. So there's tons of carbon in these rings. It's just not shown as a little C because if you put every single bit there of carbon and hydrogen, it would be really hard to read them and compare them easily. So you do need to draw this one. Okay. The important thing here is you have the two subunits, what they're called, and what they are called when they're combined together. So a glucose subunit and a fructose subunit makes a sucrose disaccharide. A galactose subunit and a glucose subunit makes a lactose. A glucose and a glucose makes a maltose. So the monosaccharide unit, the mono means one, right? And the saccharide is the carbohydrate. So the monosaccharide unit like here, a glucose, it's a single sugar ring. Those can be combined into lots of different molecules. Like sucrose here is a disaccharide with two monosaccharides making it up. And also polysaccharides. So here's a chain of several of those subunits, those monosaccharide units put together. And that's going to be examples like starch, cellulose, and glycogen. We already looked at starch and glycogen already today. Those molecules, those carbon compounds, are really important for energy transfer between organisms. But they're not the only energy transfer molecules. Another carbon compound that's really important for energy transfer are lipids. So lipids are fats. Often a group of them are called fatty acids. And do you remember how they're digested? Look in your notes. Well, lipids are digested by lipase, is the enzyme. It was produced in the pancreas and released into the small intestine. So when you have a fatty acid like this, it's a long carbon chain with a whole bunch of hydrogens on it, and then it's called carboxyl group at the end, carbon and oxygen groups here at the end. That can be broken down to get energy. Okay, next thing are the macromolecules of life. So macro means big, so the bigger molecules of life. That tend to be the ones made of many smaller subunits. And the four of them are carbohydrates, lipids, proteins, and nucleic acids. We're focusing more on these three because they're more the ones that are involved in digestion. And nucleic acids are really critical. That's the things like DNA, but they're not nearly as much numbers. They're not as key to digestion. So, based on your knowledge of health and nutrition, can you name them? Well, we already just did, but think about where you've seen them in different food sources. How do you translate this into foods? So carbohydrates, example, breads, anything with flour in it, it's going to be carbohydrates. Lipids are the fats, and proteins like meats, beans, that kind of stuff. Nucleic acids are pretty much everything that has DNA, which is every organism. So they're carbohydrates, lipids, proteins, and nucleic acids. And when we do digestion, we talk about the first three and the different enzymes that work on them. And so far today, we've focused mostly on carbohydrates. Did a little tiny bit about lipids. Which enzymes work on lipids and which enzymes work on proteins? So we already talked about lipids enzyme being lipase, and proteins have a couple. Pepsin is one, and trypsin is one in the stomach and in the small intestine. Remember, you need to know the enzyme, what it works on, and where it works. Okay. So this is a really good table because it helps connect all these ideas. So here's our four macromolecules: carbohydrates, proteins, lipids, and nucleic acids. And then we can break that down. Carbohydrates, the smallest component of that is a monosaccharide. And then we can look at examples of that. So monosaccharide examples would be glucose, galactose, ribose, fructose. They're all types of monosaccharides. Disaccharide, where di means two, is two monosaccharides combined. That could be maltose, lactose. So you drew that picture of that, the different disaccharides. And then polysaccharides are where you're taking in multiple. Poly means many monosaccharide units. So starch, glycogen, cellulose, and chitin is another one we haven't talked about yet. We're going to be focusing on starch, glycogen, and cellulose, though. The smallest subunit of a protein are called amino acids, and proteins can make up enzymes, antibodies, peptide hormones. Some hormones are made from proteins, some are made from fats. Antibodies for your immune system are proteins, and enzymes. So even the enzymes that break down proteins are actually proteins themselves. Like trypsin and pepsin, those are proteins. All enzymes are proteins. The lipids, the fats, there's different subcomponents based on what kind of fats they are. So glycerol is one. So triglycerides, the fat cells that we have in our bodies that are important, those include triglycerides. Fatty acids, those are in cell membranes. So phospholipids are examples of fatty acids. They're in cell membranes, and phospholipids are important for steroids. Some hormones are steroid hormones. They're actually lipid hormones, and we have the peptide hormones, the protein hormones. And the nucleic acids, the subunits are nucleotides, like the different bases in DNA. Remember that from our last biology unit? Adenine, thymine, guanine, uracil, and cytosine. So DNA, RNA, and ATP are instant energy molecule. So you don't need to memorize this, but you need to be familiar with this. It's really, especially taking biology next year, because this really connects all the different things we've done this year together. So we're not going to do much with nucleic acids. We're going to do a whole unit on DNA and genetics. Okay, so lipids and carbs are important for energy, but proteins, they're really important for building cells and building enzymes. And if they're for building cells, then they can build tissues, and they can build organs, and they can build organ systems, and they can build organisms. So the entire hierarchy of life is based on proteins, which is another carbon compound. And the energy to make those cells and make them function and metabolize and everything, that comes from lipids and carbs. Okay, so proteins. So here's a little full protein that's fully folded and formed. That is really made of a chain. This is a long chain of amino acids. Each little dot is an amino acid, and that chain will fold into like these helices, kind of twisted around shapes, into a very specific shape, and the shape helps it do its job. So if you're going to be digesting some protein, you have to take this protein and break it down into its chain, kind of unfold it, and then break apart the bonds between the little bits here, little amino acids, into maybe a tripeptide or a dipeptide, 2 bits or 3 bits together, and then break those down further into individual amino acids. Once I have the amino acids, then I can use them kind of like Lego blocks and reassemble them into a different protein that my body needs. Maybe I'm getting taller, and I wish I was getting taller, and I need to make some more bone cells to have my bigger, longer bones so I can be taller. So I need to take the amino acids and the protein that I eat, and then break it down, digest it completely, so I can use those amino acids to then make proteins to make my bones bigger. So in steak, for example, the main proteins are myosin and actin. Those are 2 proteins that are in your muscles of us, as well as other things that we eat, like in cows. Our body will then take those big proteins and will digest it and use the amino acids from that to then make our own proteins. Like another example here is hemoglobin. Hemoglobin is an important protein in your red blood cells. It helps carry oxygen around your body. So we take the molecule, break it down, and then build it back up to what we need. So here, for example, this is the myosin protein. It's a pretty big protein in muscles. And then we can break it down to there's thousands of these amino acids. In each branch of myosin, there's about 2,000 amino acids. So there's 20 different amino acids, and they all have different R groups on them. But they have nitrogen, carbon, oxygen, and hydrogen in them. And then something here. You'll learn more about this if you take biology next year. But the idea is we can use these blocks to make other proteins that our body needs. So here, if we start, it's the reverse process. We have the amino acids we broke down. We took our myosin from our steak we ate, and we broke it down to all the amino acids that look like this, the molecular level. And now we have them here, now represented as little circles. And I'm going to combine them into a chain called a peptide, or a polypeptide chain. And then I'm going to take that chain and I'll fold it to be a protein. And this protein is now one my body needs. Maybe for longer bones, maybe it's for red blood cells, maybe it's for an enzyme. It has some sort of purpose in the body. So I break it down to then build it back up to a different protein that I need. But the building blocks, the amino acids, the same 21s exist between almost all organisms on Earth. Okay, the last thing for today is comparing different carbon compounds. So the three main polysaccharides we're focusing on are starch, glycogen, and cellulose. So what do you guys already know about these? Well, hopefully you remember about starch is one where we consume quite a bit of it in our carbohydrates and breads and things like this. So it's a polysaccharide, right? Multiple monosaccharide units, and it's what we call a moderately branched. So it has some branches off of it, but not a ton of them. And it's produced by plants and then used in energy for energy storage. So it could be in starch granules, could be in chloroplasts, could be in roots, tubers. So potatoes have tons of starch in them. The starch in flour comes from wheat. All those kinds of things are plant sources of starch, and our body is very capable of digesting that starch and getting the energy from it. The next one is glycogen. Glycogen has even more branches than starch does, and our body can make glycogen. So when we're trying to store our excess energy, the extra sugar, the glucose, if our glucose level is too high, our liver can turn those glucose subunits into glycogen. Here's what it looks like from far away, in liver and in muscle tissue as well, to store the energy for later on when we need it. The last one here is cellulose. Cellulose doesn't have any branches, it's just this long kind of twisty chain. It's very, very long, and it's produced by plants. And most plant cell walls contain lots of cellulose, and it helps support the plants. But we learned in digestion that we cannot digest cellulose. Our bodies cannot digest cellulose. So when you eat celery, you're going to pass cellulose right through your digestive. It's really good for your body to kind of clear things out, but we can't get any energy from it. But ruminants can. So what you're going to do, your kind of comparative task, is to use this software. I'll show you how to use it in a second, a website called BioModel 3, and you're going to compare glycogen, cellulose, and starch. So I just talked about the difference between them, and I want you to use it as a practice to use the software where you already kind of know what you're looking for. Then you're going to look at some other molecules. You can pick 2 disaccharides. There's a whole bunch of options on there. You can pick 2 and compare and contrast them. And then you can pick 2 lipids, compare and contrast them. Some of the molecules you can select and manipulate. Some of them you just watch a little video of it kind of spinning around in space. It's up to you which one you pick, and I'll show you how the software works next. But you're going to make notes on these 2. So 2 comparisons you have to do, 2 disaccharides and then 2 lipids. You're going to make notes, compare and contrast, so what's similar and what's different, and make sure you submit it in your homework for this lesson so I can give you some feedback on it. If you want to look into other ones, that's great. I recommend you look into other ones, but you only have to take notes and compare 2 disaccharides and 2 lipids. So here's the software, BioModel 3. I'll attach the link to the lesson. So if you look here, you can click on all these different ones. So we're not looking at vitamins or proteins or nucleic acids. You're welcome to if you want to. We're going to focus on carbs and lipids. So if we look here, the first tab, monosaccharides, you can click on a button here at glucose, and there's a glucose. So these are the different carbons. Carbon 1, they're all numbered. People who study this in more detail. And the information here is much more you need to know for the 10th grade. Okay? But it gives you a chance to compare and kind of look at the structures. And if you look here, this one's fructose. Okay? It's different. It has 2 branches off of it, but still a single ring. Okay, so you can look at that. I can go to the next page by clicking over here, and I can look at some disaccharides like sucrose. So here's sucrose, and they've highlighted it in colors. So we have the glucose is the green, the fructose is the purple, and the bond is the red part. I can show them as their own color. So we have white is hydrogen, red is oxygen, gray is carbon. Can also look at other disaccharides. So the task is to compare 2 of them. So here I have maltose which is 2, and I have cellobios here as well. And I can again look at them, each atom for their own color as well if I want to. Okay, for polysaccharides, you've got cellulose, okay, long linear chain, no branching. We can look at different types of starches like amylose, okay, more ring-like, or amylopectin. This one has some branching, more common to starches. You can look at a little video of it if you want to see a full. There, that's the full thing. It's too big to do, so they're showing you the whole thing if you click a little video clip. You can look at glycogen. Look at all the branches on it. You can look at other ones as well, but these are the ones we learned: cellulose, starch, and glycogen. You can go back to the main menu down here, and you can go to lipids, and you can look at a fatty acid. So here's one, palmitic acid. You can look at oleic acid, and just pick any 2 you want and compare them. Right, here's another one. And the descriptions can also help you in your comparison. You can look at triglycerides, another type of lipid. You can compare this one. It's got 3 chains, different color bits. You can see that to help you. You can look at a phospholipid if you want to compare. Any 2 lipids is fine. Okay, here's a hormone, cholesterol, steroid. Take a look at that. So it's not a hormone. You can look at the lipid bilayer, so the actual cell membrane model, showing the lipids as well. You can add a water molecule. Okay, so you can go back to the menu and see all these different things. It's up to you to compare what you want to compare. The task is 2 disaccharides, 2 lipids. Okay, so your homework is to put up all the notebook pages from today. It should be 2 pages. If your comparison gets more detailed, you have a small notebook, you might need more than 2 pages. Upload all of them, and make sure you compare and contrast the 2 lipids and 2 disaccharides for me. I'll give you some feedback on that. Okay, so as always, if you have any questions or concerns, please feel free to email me, ask questions, and I hope you have a good week. Take care, everybody.